So happy this time is very strange because this time we're going to have a new start. Yeah, you look at this page. Since this week, we will have Martin Soor as our partners on this iCANX talks. So welcome, Martin. Yeah, I'm so happy and so glad to see this time all of us here to welcome Martin. And very beginning, I want to give Martin a brief introduction. So Martin was originally from Kenya, right? From Africa, and he's a superstar. He got to the school, from Kenya, and then go to US. And now he's already a professor in Iowa State University, doing super, excellent research. And last year, he became our ACX Nano Rising Star on the stage. And then now we welcome Martin. And Martin, we welcome you. Say something, to get on iCANX, please.
Thank you very much, Alice. It's a great pleasure to join you and Paul and Jagadish on this platform. It's a great platform for us to promote science across the world, and hopefully we'll get more participation from the US and from Africa. And I'm very happy to be here. Thank you very much. Appreciate it.
Okay, so Martin, today is your first show. Now I give the stage to you. Yeah, please share your stage and start it.
All right.
Yeah, all right. We start for you. Yeah, I stop sharing. Then you can.
All right. So all right, thank you, everyone. Welcome to iCANX Talks, Connecting the World and the Universe. My name is Martin Thuo, as you heard. I'm from Iowa State University, right in the middle of the US. I'll be moderating for today's session. And this month, we've had the privilege of having Professor Chen from Peking University, Professor Odom from Northwestern University. And today, we have the honor of having our young scientist, Professor Balla Diop Ngom from UCAD in Senegal, and Professor Deblina Sarkar from MIT. We will have the whole panel after the discussion. So for the first part of the talk, I want to introduce Deblina. And Deblina is a professor at MIT and is a rising star in the area of nanomaterial, and she will be sharing with us some of the work that she's doing at MIT. She has many awards, including being named top inventor by Technology Review. Her PhD thesis was ranked in top 3 in North America, which is amazing. She's also a professor in MIT Media Lab, without spending more time going through all her accomplishments and award, I want to welcome this Deblina to the stage. Deblina.
Thanks a lot, Martin, for the kind introduction, and thanks, Alice, and thanks everyone for having me here today. It's a great pleasure. So I'll go ahead and share my screen. Okay, you can all see my screen, right? Okay.
Yes.
Okay, great. So I'm actually a new assistant professor at MIT. My lab is undergoing renovation, which is very exciting. And so I started my career as a nanoelectronics researcher. And then I did a steep transition to neuroscience for studying the brain. So what I will do today is tell you the story of my journey from nanoelectronics to neuroscience, and tell you about the amazing synergies in these two fields. And I will do that by sharing with you one example of project from each of these fields. So let's get started. So all of you must have noticed how your laptop heats up after working for some time. The heat generated is so high that a while ago, I even cooked an egg on my laptop. So let us understand where this heat comes from. So your computer has a billion switches in them, and in those switches, the electrons need to jump over an energy barrier to cause current flow. So when this barrier is high, very few electrons go from source to drain, and your switch or transistor is off. And when you want to turn on the transistor, what we do is we apply a voltage to another terminal called gate, and you reduce the barrier height. So now more electrons can go from the source to the drain, and your transistor or switch can turn on. However, this way of switching has a fundamental limitation as to how much power this transistor will need. So ideally, you would think that this transition from off to on state will be very steep. That means with application of very small supply voltage, you will be able to turn this transistor on. However, we do not live in an ideal world. And in an actual situation, what happens is that the current increases slowly with voltage. That means you need a sufficient amount of voltage to get a significant on current to have a good on to off ratio. And this steepness of this curve, that is how much voltage you will need to have a certain increase in current, that is defined by a parameter called subthreshold swing. And that's basically the voltage required to change the current by 1 decade or tenfold. So what happens when you want to make the transistor smaller and smaller, and you want to make the transistor smaller so that you can fit in more and more of this into a single chip, and then you can increase the functionality of the chip. Now, when you try to make this transistor smaller and smaller, what happens is that the electric field from the drain kind of encroaches into the channel region, and that decreases the barrier height in the source side. That means these electrons, they will go from source to the drain even when the transistor is supposed to be off. And that would increase the off-state current and basically the off-state leakage and the off-state power. So as you can see in the curve here, the blue curve is for a long channel transistor. You can see the steepness of the curve here is good. That means the subthreshold swing is smaller. The steeper the curve, smaller the subthreshold swing, because it's the voltage required to increase the current. And when you make the transistor smaller, because of this effect, which we call a short channel effect, the leakage current has increased, and you can see the subthreshold swing has degraded. Now, in the world of electronics, the rule of thumb you can take as thinner can be smaller also. That means if you make the channel thinner and thinner, it can also be smaller in its length. So how thin can we make our channel? You can actually make them atomically thin. So you can make the channel only few atoms in thickness. That means it can be 10 to 100,000 times smaller than the thickness of your hair. And for that, we can use 2D semiconductors. 2D because its thickness is so small that this third dimension of thickness doesn't almost exist. It's only two-dimensional, that is just in planar dimension. You can use 2D semiconductors, and one such kind is transition metal dichalcogenide. So transition metal dichalcogenides, they are actually layered structures. So within each layer, transition metals and chalcogenides, they are bonded covalently, but between two layers, there is weak van der Waals bonding. So that means you can peel them off layer by layer. And each layer of this material can be extremely thin. For example, molybdenum disulfide, which is one of this 2D transition metal dichalcogenides, a single layer of it can be only 0.65 nanometer in thickness. So you can solve this problem of short channel effect, and you can make the transistor smaller and smaller in length by making these channels with atomically thin 2D materials. But does that solve all the problems? That solves the problem to some extent, but not all. This is because as far as the power goes, we have a fundamental issue here. And I can explain it with this schematic here. So suppose at the off state of the transistor, you have a certain barrier height of H. But when you apply a gate voltage of VGS, say the maximum amount by which you can lower the barrier would be similar to VGS. You cannot do better than that. And assuming this is an unit in electron volts. So that's the best case by which you can reduce the barrier if you apply a gate voltage, if you apply a certain gate voltage of VGS. And since the electrons in the source, they are distributed according to the Fermi-Dirac distribution, and which you can approximate for Boltzmann distribution, they will go from source to channel, and the current will increase following this equation, exponential VGS by kT, where K is a Boltzmann constant and T is the temperature. And if we calculate from here how steeply the current can increase when you apply a certain voltage at room temperature, that will come out to be 60 millivolt per decade. That means is that to increase the current by tenfold, you will need at least 60 millivolt of voltage, and you cannot do better than that. And so what does this mean for our transistor design? So that means in the very best case of the transistor, the steepness is 60 millivolt. So that means if you want a current on to off ratio of say 10,000, that is 10 to the power 4, you need at least 4 multiplied by 60, 240 millivolts of voltage to get that on to off ratio. Now that's the very best case. But normally the transistor, the subthreshold swing will be even worse. In that case, you would end up needing even higher voltages. But as you know, the power is, square of the voltage. So basically the power increases as you, keep on having to need more and more voltages to turn the transistor on. So if we really want to reduce this power, we have to design transistors which are much steeper, which can turn in much more, with much more much lesser voltages. And so you have to design transistors where the subthreshold swing is less than 60 millivolt per decade. But the 60 millivolt actually comes from this very fundamental distribution, thermal distribution of electrons. So that means just using a new kind of material will not help us to overcome that limit. We can maybe get this best case, but if we really want to overcome this fundamental thermal distribution of electrons, we have to think of fundamentally different carrier transport and not just a new material. And so you can think of, instead of making the electrons jump over the energy barrier and be very energy expensive, can you make the electrons just sneak through the barrier like this, just like a ghost walking through the wall? Now you must be thinking, how is this possible? Because we cannot just walk through a wall like that. So while classical physics says that it's not possible, quantum mechanics says that it is possible, and this is because of the wave nature of electrons. And so it is possible for them to just sneak through the barrier, and the scientific name for this is quantum mechanical tunneling. So that's great, but we still have some problems that we have to solve. Suppose there is this person here who is very low power loving and wants to just tunnel through a energy barrier like that. But by the time he reaches the other side of the barrier, he would look something like this. And this is because the electron waves decay exponentially as it passes through the energy barrier. So that reduces the current significantly, and such that it's even extremely challenging to even turn on currents if we use this quantum mechanical tunneling effect. And the current often remains below the noise level. So if we want to design transistors using this tunneling phenomena, which we call tunnel field effect transistors, we'll have to think of very intricate device design. So if we want to improve this and make sure that these waves do not decay that much, we have to make a design in which this energy barrier height and width can be reduced simultaneously, and you need to have very high electric field at the tunneling junction. And all these things have to happen at the same time. Moreover, note that having a steep subthreshold swing for a very small region of current will not help to reduce the supply voltage. If we really want to have a low supply voltage to turn on the current, you need to have this steep subthermal subthreshold swing over a large range of current. And the International Technology Roadmap for Semiconductors, it recommends that we have this steep subthreshold swing for at least 4 decades of current, which has been challenging to obtain. So next I will discuss the transistor that I had designed, which helps us to overcome these challenges and attain subthreshold swing that is subthermal and overcome this fundamental limitation in power. And the transistor, we call it the Atlas DFIT, which is the short form for atomically thin and layered semiconducting channel tunnel field effect transistor. So in the next few slides, I'm going to explain how this transistor works. So in here, we used 3D material as the source, and the channel is 2D in nature. So we use 3D material as a source so that we can still leverage the mature doping technology of 3D materials to design a highly doped source region. And high doping in the source region ensures that we get high electric field at the tunneling junction. The channel is still 2D in nature. So the channel is atomically thin. So as I mentioned, you have to have extremely thin channel if you want to improve the electrostatics. That helps us to make the channel length smaller and smaller. Remember, thinner the channel, smaller it can also be in its length. So that's why the channel is still 2D in nature. And the heteros junction between this 3D and 2D material is van der Waals in nature. So there is no chemical bonding between the two. So that will make sure that there is no diffusion of atoms between these two layers. So you can have a very steep tunneling junction. So with this design, we can have high electric field at the tunneling junction. But at the same time, we should also try to reduce the barrier height and the barrier width. So how do we do that? So to reduce the barrier height, we can, basically the energy barrier height, what we can do is we can judiciously choose the different materials. So in this case, I have chosen germanium as the 3D material and molybdenum disulfide as the 2D material. So these materials are chosen because among the most commonly known 3D materials, germanium has relatively low electron affinity and low band gap. And molybdenum disulfide has slightly higher electron affinity. And when you align the bands, we will see that the height of the barrier, which is defined by the energy difference between the conduction band of molybdenum disulfide and the valence band of germanium, as shown by this red arrow here, it comes out to be small. Now there has been a lot of discoveries of new 2D materials. So it is possible to even improve this barrier further and barrier height further and optimize our design based on, mixing and matching different kinds of materials. So there is a lot of opportunity of improvement. So we have high electric field at the tunneling junction. We have low barrier height. How do you also reduce the barrier width? So for discussing that, let me explain the band diagram of how this transistor works. For this, I'm drawing the band diagram across the z-axis here. And the z-axis, it goes from the germanium source to the 2 layers of molybdenum disulfide. And then we have the gate dielectric, and then we have the gate metal. So you're drawing the band diagram across this line. And you can see that when in the off state of the transistor, the field regions in germanium, that the region in energy which are filled with electrons, they do not coincide with the empty regions in molybdenum disulfide. So we have minimal current here. If we go higher in energy, we encounter the band gap of germanium. So, this is a forbidden gap, so electrons do not stay here. So again, minimal current or no current. And then when you go higher in energy, we encounter the conduction band of germanium. Now we have doped this germanium p-type, so the Fermi level is in the valence band. As the number of electrons decays exponentially, very few electrons are there in the conduction band. So again, the current is low. And when you want to turn on the transistor, what you do is you apply a voltage to the gate, you bring all the bands down. And in that case, the filled regions in germanium, they are actually coinciding with the empty regions in molybdenum disulfide. So, a lot of electrons can flow from the 3D source to the 2D channel. And so current increases steeply. And this is the steepness basically is attributed to the fact that because of the presence of the band gap, it cuts off any leakage current that you would have otherwise got from this region. Because it's the forbidden gap, no electrons is there. So we have basically cutting off the Fermi tail from here. Now, as you can see here, the width of the barrier, that the width through which these electrons have to tunnel is given by these 2 layers of molybdenum disulfide. 2 layers of molybdenum disulfide is extremely, extremely thin. So each layer is 3 atoms in thickness, 2 layers is 6 atoms in thickness. So the tunneling barrier can be made atomically thin, in this case, only 6 atoms. So here is, the TEM image, and you can see that we have the germanium and we have the 2 layers of molybdenum disulfide. But what you see here also is that between the germanium and the molybdenum disulfide layer, there is a thin layer of germanium oxide. Now this is a challenge because anything in here would increase the tunneling barrier width. But this is a challenge, but also an opportunity, because this means in future designs, it will be possible to remove the germanium oxide and passivate the surface and then improve the tunneling current even further. So here I'm showing the subthreshold swing characteristics, and here the red line marks the fundamental limitation of 60 millivolts per decade. And as you can see here, for a conventional field effect transistor, which is marked by these green triangles here, the subthreshold swing, the conventional transistor actually marked by the blue squares here, that in this case, the subthreshold swing is always above this fundamental limitation of 60 millivolt per decade. But our Atlas TFET, which is shown by these green triangles, actually overcomes these fundamental limits and gives a minimal subthreshold swing of 3.9 millivolt per decade, but also an excellent subthreshold swing, which when averaged over 4 decades of current is almost 30 millivolts per decade, which is almost half of the fundamental limit that can be achieved. Now, apart from having the lower subthreshold swing and overcoming this fundamental limitation of power, this device is made up of, as I said, 6 atoms thin channel. So apart from power scalability, this device can also allow simultaneous channel length or dimensional scalability. So it might be possible to achieve both power and dimensional scalability in this same device. So during my, during this research, while I was working on low power electronic devices for electronic computation, I also got interested in other low power electronic systems, especially the brain. So the brain, which is this amazing, organ of our body and is a boss of our body. So it just weighs 3 pounds. But it's really the brain which distinguishes who we are and separates us from other animals. And if you look inside the brain, you will see that it's formed of a dense, jungle of, and it's formed of hundreds of billions of computational elements called neurons, and which talk to each other using electrical and chemical signals. So the brain does all this computation, but consumes only about 20 watts of power, similar to a mere light bulb. So however, at present, we know very less as to how the brain works. How is it so energy efficient? So why is it so difficult to understand the brain? So if you think of electronic circuits, we design them ourselves. We know all the interconnections. We know not only the major components, but you also know how they are connected to each other. And in each component, you know the physics of semiconductors, metals, insulators. We can draw band diagrams to explain them. However, in the brain, Mother Nature has made it so complicated, so intricate that we human beings have not been able to decipher that complicated interconnected circuitry, or even know, like within each small element, how the biomolecules are arranged to lead to its functionality. And if we see inside a small region of the brain, we'll see that it's formed of a dense jungle of biomolecules, which will probably look as similar to the gum wall in Seattle. So if we really want to decipher this dense jungle, we will need some super resolution technologies. However, the present super resolution technologies are extremely expensive and needs expert handling. So in this case, a technology called expansion microscopy is very interesting. And in expansion microscopy, what you do is, for example, instead of using complicated hardware to see small features in the sample, we expand the sample itself. So suppose there are 2 points which are very close together, and they cannot be resolved by a conventional diffraction limited microscope. By physically expanding them and separating from each other, it is possible to resolve them now. But how do we physically expand a sample? So you can do that by basically using hydrogels. So hydrogels are materials which can expand and swell up when you add water to it. So what you can do is we can take a piece of brain tissue and then we embed in that hydrogel material, and then we just add water. So the brain tissue expands along with the hydrogel in this case. And this, what you're seeing in the image, is the same tissue before and after expansion. So in the first round of expansion, we achieved around 4.5-fold expansion factor. So that means if we have a diffraction limit of the microscope is 300 nanometer, by 4.5-fold expansion, we can get around 60 to 70 nanometer resolution. With the same hardware, that's the same diffraction limited microscope, we can overcome this diffraction limit. So to tell you a little bit about how this technology works. So we start with a fixed biological specimen, and then we anchor the biomolecules of interest that we want to, see in great details. Then we form this swellable gelling network and connect these biomolecules to the swellable gel. And then with a mechanical disruption step that you have to do to overcome this physical binding and the tension in the tissue, then we add water, and then the gel expands, and it expands the biomolecules along with it. You have done a lot of characterization to make sure that the expansion is isotropic in nature. Now we can think that we can also do this process iteratively. That is, after first round of expansion, we can iteratively expand, and then we can get higher expansion factor. And then higher expansion factor means that we can get higher resolution. In the first round of this iterative expansion microscopy, which we call IEXM, we had to use DNA handles. So in this case, DNA was used as a proxy, basically, to know the location information of the biomolecules, but the biomolecules themselves were lost after the expansion. So to overcome this challenge and to retain the biomolecules over multiple rounds of expansion, we developed a technology which we called expansion revealing. And the advantage is, of course, apart from the fact that you retain the biomolecules and you can use off-shelf antibodies to, target and visualize your biomolecular nanostructures. It also has this advantage that the effective size of the antibodies that we can use in this case basically gets reduced by the expansion factor. So if the size of the antibodies are, say, primary and secondary together around 30 nanometer, then by expanding about 20-fold, the effective size becomes around 1.5 nanometers. So that means it is possible in the future to get to that kind of expansion level, and the error will still be very less. Another novel effect of this technology is the decrowding effect. So suppose there is a target biomolecule, which is this red one here that we want to attach to the antibody. But because these biomolecules remain in a very crowded state, this antibody cannot reach this biomolecule of interest. But when we expand and separate out basically these biomolecules from each other, now they can, this antibody can target this red biomolecule here, and we can get the staining and visualize this nanostructures. So that is why you see that there are many invisible structures which could not be discovered with other technologies, but that have been revealed by our technology. That's why we call it expansion revealing. And I will show you one example of how what kind of invisible nanostructures we see. And apart from just seeing protein molecules, this technology can be used to look at nanostructures of RNA, DNA, lipids, basically any kind of biomolecules, you name it. So we tried to, look at stereotypical structures and see how our technology works. So we looked at microtubules. So microtubules are structures through which, basically, they are platforms on which motor proteins actually walk through on these microtubules. So I thought it was very fascinating, coming from a nanoelectronics background, that there are actually protein molecules which have almost like two legs, and they are, walking around carrying cargo. So these are hollow structures around 20 nanometer in diameter, which we cannot resolve using a conventional microscope. But when we use expansion revealing technology, we can see that we can decipher the two peaks, that is these peaks of that you get from these hollow structures if you draw a line here. So I wanted to look at, the nanostructure of synapses, which is a central structure in how one neuron communicates with another neuron. And here is an image of a single synapse and showing two different biomolecules, which occurs in two different neurons across this transsynaptic cleft. And you can see that without the EXR, they look like overlapping blob. But after EXR, you can actually separate them out and see their nanostructures much more clearly. So I wanted to see what kind of biological questions you can answer with this technology. And for that, we collaborated with neuroscientist, Professor Thomas Glanville, and we wanted to look at whether there is an organization between the synaptic structures, or are they, randomly oriented. If there is an organization, what does it tell us about information processing in the brain? So we looked at three different biomolecules here. One is a biomolecule called RIM, and basically that RIM dictates where the synaptic vesicle is going to be released and going to release the neurotransmitters. So basically, to tell you very quickly about how the brain works, like how the neuron communicates with another neuron. When one neuron becomes active and wants to talk to the second neuron, what will happen is that the potential inside this first neuron increases, and there are voltage-gated calcium channels which will open up. So the calcium ions goes in, and it leads to the fusion of these neurotransmitter containing vesicles. So at that point, these neurotransmitters will be released, and they travel through the synaptic cleft, go to the next neuron, and get attached to the receptor molecules there. And then receptor molecules, when they get attached to these neurotransmitters, they also open up, and then they let in the flow of other ions. And as they let in the flow of other ions, the potential inside the second neuron can also change. So that way, one neuron can influence the other neuron, and that's how they talk to each other. So this molecule RIM, which is shown in red here, dictates where the synaptic vesicle is going to fuse. We looked at another molecule called PSD95 that acts as a scaffold for these receptor molecules, and then the calcium ions. And what we found something very interesting, and this is basically what you are seeing is 3D view of these proteins RIM and PSD95. And what we found very interesting is that these biomolecules are just not randomly distributed, uniformly distributed, but they form non-uniform nanoclusters. And what is even more interesting is that nanocluster of one protein, say RIM, they are colocalized, but basically they are located just above each other, and the centers of these nanoclusters, they're aligned with nanoscale precision. So not even micron scale precision, the precision is nanoscale in nature. So this is very interesting. Why should, first of all, they form nanoclusters, and why would the centers of two different proteins, which are occurring in two different neurons across the synaptic cleft, be aligned with such nanoscale precision? So this can be actually explained in the following way. As I mentioned, that RIM is the protein which dictates where the synaptic vesicle is going to fuse, and PSD95 occurs, basically acts as a scaffold for these receptor molecules. The fact that these receptor molecules are clustered just above the region where this synaptic vesicle is going to fuse, that reduces the distance that these neurotransmitters have to travel. Because if these receptors were just randomly oriented, these neurotransmitters had to travel longer distances to reach the receptor molecule. And so by aligning and this and nanoscale precision, Mother Nature has designed to increase the efficacy of neurotransmitter travel, and that increases the efficiency of information travel, information processing in the brain. So initially, I showed an example that you can reduce the barrier width in our tunneling field effect transistor by using atomically thin materials. And as nanoelectronics engineers, we worry about nanoscale precision, nanofabrication, and all those things. But when you come to know that in our brain, Mother Nature has actually arranged biomolecules with such nanoscale resolution to improve the information processing in the brain, that really blows our mind. And we can actually not only look at healthy brains with this technology, but also different kinds of neurological diseases. And one interesting structures, one kind of interesting result that we found is that in Alzheimer's brain, there are these misfolded proteins, but we see that they are forming periodic structures within the brain. And these periodic structures are actually not visible with other technologies, but we can only see when we apply this expansion revealing technology. So when we talk about brain diseases, we call it brain disorder because we think disorder is associated with diseases, but we found in here, to the contrary, there's order and periodic structures which can also be related to a disorder. So we can actually expand to up to 100-fold currently. So you can blow out the brain 100-fold to look at different kind of structures with higher resolution. So, to summarize, I showed you, two different technologies, a transistor, which overcomes fundamental limitation in power, and then another technology, which helps us to look at the biomolecular building blocks of the brain. So two completely different technologies, but you can also see some interesting synergies between the two. So in my own group, basically, I'm trying to fuse these two fields together. And the idea is to develop, technologies for novel nanoelectronic computation, but also, fuse them with biology to create new paradigms for life-machine symbiosis. So we are developing, different kinds of novel bioelectronic hybrids, which can work at the nanoscale, and also nano implants for energy harvesting and wireless sensing. And we, of course, continue to push forward the boundaries of, low power energy efficient computing. So I do have a lot of openings in my group for students and postdocs in various fields of, applied physics and nanoelectronics. Specifically, there are two postdoc openings, actually, one for brain stimulation and recording, someone who has experience with electrophysiology as well as, brain stimulation. But another opening is for a postdoc for working with magnetoelectric devices. And so if you're interested, you can shoot me an email, and more details can be found in this website here. So finally, to thank all the wonderful people, my students, postdocs, my colleagues, mentors, whom I have worked with, and without whom this work would not have been possible. And of course, also to thank all my family members who have been extremely supportive. And finally, thanks to you for your kind attention and for being here today with me. Thanks a lot.
Thank you very much, Professor Deblina. Really appreciate that wonderful talk. I have a few questions from the audience. One of the question is from Yungqing from Wuhan. Thanks for your wonderful talk, Dr. Deblina. Do you want to touch quantum computing? What is your vision of the field?
Yes, actually what we are doing is, I would say, something in the midfield between quantum computing and conventional computing. So we are using quantum mechanical effects, but it's still like the transport is still, I would say, classical in nature. Quantum computing, I think, has great future. Right now, we still have to overcome some limitation in terms of making them work at room temperature. But I think before even we go there, there are a lot of innovations and intriguing things to do, even at room temperature, where the transport is still, like not quantum, but we can still overcome and, get beyond the Moore's law. And so we're basically exploring that area right now.
Okay, very good. The other question, maybe I can chime in, is how do you manage the interfacial properties between your Atlas TFET so that you don't have adventitious contaminants that will interfere with your tunneling?
Oh yeah, that's very interesting because that's very important, because anything at the interface can basically kill the devices. So, I mean, there are device processing. Of course, you work in the clean room and make sure that you clean the interface when you work with germanium. For the 2D materials, also, we used a CVD process to get the 2D materials, because we found that if we were using this exfoliated way, exfoliation, to get the 2D materials, we're getting a lot of contamination from basically the gum that we use for exfoliation and things like those. So yeah, we have to make sure the interface is clean, and by using CVD materials and the transfer process that we do at each case, and looking at ACM at every step, we make sure that we are not contaminating.
Okay, and do you have any issues with the induced gap states with such proximity to your materials?
Induced what? Sorry.
Induced gap states because of the difference in
yeah. So the thing is, one interesting property of the 2D material is that their interfaces basically are, I would not say like completely defect free, but are relatively better compared to 3D materials, because between each layer, between the layers, there are no covalent bonding. So their interfaces are saturated, so there are no defect states for the 2D material, but the 3D material though still has a defect states. So between the 3D and the 2D material, there are defects, and so we'll have to passivate the 3D material for the 3D to 2D material interface. But what is good is between the 2D material and the gate dielectric, the interface can be relatively pristine, so the electrostatics can be much better. And yeah, if there are defect states, that can kill the tunneling.
Very nice. With that, I want to thank you very much for your wonderful presentation, and congratulations on the Young Scientist Award.
Yeah, thanks, Martin.
Thank you. All right. The next speaker we have today is Professor Balla Ngom from UCAD, University Sheikh Anat Diop, and Professor Balla has an interesting academic training. He started out as a physicist, and then he did his bachelor's in UCAD, then did his PhD back in Senegal, and then he went to South Africa for his second PhD, and then from there he was a postdoc at the National Research Institute in Canada. Then he went back to South Africa in the Iterative lab, working with Professor Mazer, where he did his second postdoc and third postdoc at the UNISA, University of South Africa. He then moved back home to Senegal, where he's now an associate professor. He has done so much that I'll take over too much of his time if I were to go through everything that he's doing and all the international collaborations he has. So I want to hand this over to Professor Balla. Please, welcome.
Thank you very much, Martin. Can you hear me?
Yep, we can hear you well.
Okay, wonderful. So thank you. I would like to take this opportunity to thank the organizers of this exciting talks. It is really a scientific, informative event. And thank you once again. Just would like to talk to you about today about how to develop future nanomaterial derived from biomass for energy storage application as the current energy crisis that the world is facing right now. We think that it is important and it is challenging, and it's important to look at into new material for development of new energy storage devices. Before that, in addition to the introduction, I would like just to talk about a little bit of where I'm coming from. I'm coming from Africa in the western side, a country that is called Senegal, having a population of 16 million, which is where the economic is based on agriculture. You will see exactly why we are interested in biomass, which is an agriculture residue, and transform it and trying to look at how to valorate into high technology application. So Senegal is very rich in culture. It is also the main activity is, as I said earlier, is based on agriculture and fisheries. And I couldn't believe that the best fish could coming from Senegal. And if you have the chance to visit, it is the opportunity to test the best fish in the world. And we have a lot of tourism where we have the slave trade, which is happening between Africa, Europe, Africa, and the United States. And besides that, as I said earlier, in Senegal, especially in Africa, in the world, we are facing this energy crisis. As you could see that the map, as I'm showing, just as an illustration of what is happening currently in the energy landscape in Africa. And this, we're still facing this problem for students to have access to electricity, especially in rural area, where studying is becoming a nightmare currently. And you would believe in 2021, we're still having this situation in the rural area in Africa, especially here in Senegal. And in addition to that, and globally speaking, the overuse of fossil fuel is driving the world into crisis, especially in the environmental aspect, where we're having this greenhouse effect that really changing as we've seen what is happening currently in the world, the flooding around Europe, and especially also here in Africa. We still need to look into alternative energy resources. And we believe that renewable energy could be one of the key solutions to overcome and also to go fully into the energy transition. And we believe that solar energy, especially in Africa, and wind energy, as well as water-based energy, could be one of the best solutions, especially we believe also biofuel derived from plant biomass and so on could be one of the best things for Africa. And as I said, Africa is having a very sunny and very windy continent, and it's having a lot of water. And we believe that once we focus on working on renewable energy, we could meet this transition energy and could be one of the solution for Africa crisis. And so people could say that there is a problem of efficiencies when talking about renewable energy, but going into an hybridization or using the different source could be one of the answers, beside the fact that it can't provide the full-time energy, which is depending on the weather. And the alternative solution could be looking into these energy storage devices. And one of those energy storage devices we could call up what we call the batteries. Batteries and supercapacitors, which are the main used and with the main feature of energy storage that could, in addition to renewable energy, could answer this fluctuation of renewable energy, which is due to the climate. And if we concentrate especially on supercapacitors, we could see that supercapacitors could be used in different applications such as transport, hybrid buses, wind turbine, and also transportation, smartphone, and so on and so on and so on, especially to boost the use of renewable energy. So coming into the fundamental of supercapacitors, we could see that based on the mechanism of storage, as it is a storage devices, based on the mechanism, we could see that can be divided in two type of devices. What you call electric double layer capacitors, where the storage mechanism is an electrostically based mechanism, and the most used material are carbon based, and different nanostructure have been developed to enhance the supercapacitor EDLC supercapacitor efficiency, such as graphene, activated carbon, and carbon onion. And they have the advantage of having high specific area, surface areas, and also they have also the good electrical conductivity, which is one of the good best parameter that could enhance the efficiency of supercapacitors. And they have, and in result, the results could be a moderate specific capacitance. On the other hand, we have what we call Faraday capacitor or supercapacitors, where the mechanism is a charge storage, electrochemically charged storage devices. And the material that are used in this device type of supercapacitors are conducting polymers, oxide, and also metal oxide, as well as metal hydroxide, and several oxidation states are one of the key parameters that are really needed and that are the parameters that are controlling the supercapacitor efficiency, and that is lead to high specific capacities. So beside these two, we could look to see that supercapacitors are lacking from energy densities, and they are having a high power, but trying to look at how to develop an hybrid device that automatically could, at the same time, taking into account the high specific energy that the batteries are having and the high power density that supercapacitor are having, could be one of the answers of having a best device for supercapacitor application, what we call an hybrid supercapacitors. And the answer could be a material science project, and we believe that working into composite material could be an answer to reach the best supercapacitor device that we are targeting. So to show a little basic on the important parameter of supercapacitors for us, for the audience, especially the students who are following this talk, we are targeting to look at the specific capacitors, which is in watt-hour per kilogram, the specific power, which is a watt per kilogram, as well the charge balance, specific capacitors, and specific capacities. These are the main parameters that we are looking into supercapacitor, and we would like to enhance. And our work is just based on these 5 parameters that we are interested into. So, as I said earlier, we are interested in developing biomass-driven, sustainable and innovative composite that we will be used into energy storage capacitors devices as electrode. We start our work by looking at the sustainable development of vanadium pentoxide carbon composite, which are derived from hibiscus sabdariffa. And hibiscus sabdariffa is a plant that we grow here in Senegal. It is used also as a drink. We could use especially the light red one. The light red one is a commercial drink that people are using, but the dark red one is too strong, and it's not really good for health. And we have the white hibiscus sabdariffa, which is also used especially on the food market as well. But we believe that looking at the residues of these could be lead us into devices that are green and safe for energy storage and storage application. What we do first is to take vanadium metavanadat and to look and to make it ammonium metavanadat, and we use it to produce VO2 powders. And we use also, on the other hand, we have this extract from hibiscus sabdariffa, and we mix it, the two of them, and using a hydrothermal synthesis into an autoclave, we could follow, and we have the final product, which is vanadium pentoxide and carbon composite, where we're trying to characterize. And the first characterization that we have done into this is to look at the structure of the material that we produce from this hibiscus sabdariffa. And the first, after the figure A that I'm showing is the X-ray diffraction using the three different hibiscus sabdariffa, the white one, the red, the light red, and the dark red that we have. We could see that we still have the vanadium pentoxide as well as the carbon aspect, which is just not appearing here, but we'll see that it is appear on the flowing characterization. And looking at the grain size, we could see that the grain size is depending on the type of the hibiscus sabdariffa that we are using. So and the structure of the V205, that is an orthorhombic structure, with the latest one that I'm showing currently right now. So to further go into the characterization and check exactly what we have produced, if we still have the composite that we really would like to produce from the hibiscus sabdariffa residues, is that looking at the Raman, which is in figure A, we could see that the Raman is just the peak that we are having here, the vibration mode that are coming from V205, which are completely below 1000. As we know, the Raman signal of oxide are always appearing below 1000 centimeter minus 1. So if we zoom at between 1.5 to 3000, we could see that there is, in the blue one, in figure B, the blue one is just the carbon, the D band of the carbon, which is a signature that the carbon is incorporated into the V205. To further investigate the presence of the carbon as well as the vanadium pentoxide composite, we look at the Fourier transform infrared spectroscopy of the different samples, and we could see that the bonding between vanadium oxide and vanadium are appearing here, which is a signature that we are having the vanadium oxide, and the band absorption band, which is due to the carbon-carbon bonding, which show exactly into the composite, we are having the carbon, which confirm exactly what we have seen into the Raman, as well as what we have seen into the X-ray diffraction spectrum, even if we were not able to see the carbon with the diffraction techniques, X-ray diffraction technique. So looking at the scanning electron microscopy graph images, which is one of, as the morphology is one of the key parameters for energy storage application, because we could say that the electrolyte and the active material, their interaction, the wettability is one of the best parameters that control the efficiency, the energy storage of the device, of the electrode. So looking at the ACM, A and B and C and D, so the high magnification, which has A, B, A, C, and E, and the low magnification at 200 nanometer, which is B, D, and F, are showing the different morphology that we're really having when we move from one hibiscus, the white one, the light red and the dark red one. And you could see that we are having a 2D material, which is which confirm exactly, which could be one of the morphology that could enhance the electrical property of the material, and we see exactly what we do. So as I said, we are interested on the electrochemical property of the material. Then we took this material and we have produced a device which we first look at the as a positive electrode, what the material is have been responding, and looking at the CV, galvanostatic charge and discharge, and also to extracting the specific capacity versus the specific current, we could see that the mechanism, the charge mechanism, could see that we are having a Faraday Capacitor behavior as it is an oxide, and also the electrolyte that we are using is KOH, 6 molar KOH, and we could see the charge also as the nonlinearity of the charge and discharge mechanism could confirm exactly what we have as a mechanism, which is confirming the CV measurement that we have here. So looking at what the specific capacity, we could see that we are having 49 milliampere per gram, which is the result that we got from the best result that we got from the hibiscus sabdariffa. And what we have done is that we have constructed a device using this VO2, V2O5 carbon red one, the dark red one, which we found that was the best one, and as a positive electrode, and activated carbon from peanut cell as a negative electrode in 6 molar KOH to fabricate the device. And we could see that the device have, looking at the Coulombic efficiency of the device, if we could see that we're having a Coulombic efficiency of 58.5, something like, and 86 capacity retention, which is showing the stability, the best stability that our device is having. And finally, we measured the energy density of 33.4 watt-hour per kilogram, as well as a power density of 670 kilowatt per kilogram, which is showing a potential that these electrodes, which are derived from hibiscus sabdariffa, could be used in the supercapacitor device as an electrode matter. So in order to enhance the energy density as well as the power density, as we are looking into balancing the energy density and the power density, because supercapacitor, as I said earlier, are having this supercapacity, are having this high energy density, but they are really lacking high power density, but they are really lacking with energy density. We need to enhance the energy density without compromising the power density. So we are working into balancing. So we took graphene form and we imbibed it into the material that I just presented just now, to see exactly if what could be the effect of this graphene forms. So we have used the same process for the synthesis of vanadium pentoxide, carbon, and graphene form, to look at exactly what will be, as I said, what will be the effect of this graphene form into the electrode material. So looking at first the characterization to see exactly what we have produced, the X-ray diffraction, which is in figure A, show the orthorhombic vanadium pentoxide and as well as the carbons. This time we are able to see exactly the presence of 002, which is coming exactly from the graphene, as we loaded different amount of graphene into the vanadium pentoxide carbon composite. So looking at also the Raman, which confirmed exactly what we previously obtained on the X-ray diffraction, which is the oxide part, as well as the carbon aspect part, which is appearing, which confirm exactly that the presence of graphene is really embedded into the vanadium pentoxide carbon material, and which is also further confirmed by the FTIR Fourier transform infrared spectroscopy. So looking at the morphology as the SEM, we could see as well that the FEM, which is completely different from the images that the structure, the morphology that we had previously before embedding the graphene into the composite. You could see that the morphology has completely changed, and this compacticity, but the morphology is more compact when we embed 40 milligram of graphene than 20 milligram of graphene. We could see exactly what could be the effect on the electrochemical property of the material. So we look at also the PET to be able to see how the material is nanostructured, and we could see that this is the surface area of the material, which increase. The best surface area that we could found is when we imbibed, we mass loaded 20 milligram of graphene, and we could see that we are around 20 millimeter square per gram at 20 milligram of graphene form. So looking at the nature of the pore size and the volume of the pore, we could see that we are having a pore size comprised between 2 and 4 nanometer, which is a mesoporous material, which could be a best material for electrochemical. It will allow the interaction between the electrolyte and the active material. So we have done the electrochemical CV. Here is just to compare with the activated carbon that we used it as a negative electrode, and here we are having the composite, and you'll see that the redox and the redox peaks that, and that oxidation and the redox that we're having here, which show exactly that we're still having a supercapacitor response mechanism on the material. Even if we imbibed this carbon-based material, as we know, the carbon-based material are responding in EDLC form, as the activated carbon is showing no oxidation or no redox peak. And this is confirmed by the potential discharge behavior, as the nonlinearity of the discharge is confirming the supercapacitor's behavior of the material. And all this turned into an electronic energy density of 55 watt-hour per kilogram, as we are moved from 33.5 to 55. So you could see that how the graphene that we incorporate into the matter has enhanced the energy density of the electron, even if we have a energy power of power density of 770 kilowatt hour per kilogram. So this is showing how it is important to move from biomass material and drive into high technology material that could be really important for future development on high technology, which is a green process. And looking at the stability and how the material is, we look at the coulombic efficiency as well as the capacity retention, we could see that we are having a capacity retention of 99%. So, and 99% for coulombic efficiency. This is show exactly how it is stable and how the material is really and really important. And this corresponding into a floating time of 23.6 after long time retention after the device have been ruled a long time. And if we look at the resistance equivalent resistance series of the material, we could see that at 20 after retention, the material is becoming more or less resistant, which is showing how this material, the importance of having graphene form into this composite could also enhance the property of the material. So this work is just a work that we started just 2 years ago to look at exactly, as I said earlier, how to really use this residual that are harming the environment, and also to reduce the greenhouse process, and also to boost the energy transition that we are facing right now especially in Africa, in order to solve the problem of energy. And this show also how it is important to look at the way that we develop our process, and it is possible to have a solution for our own problems, especially African facing crises, and using agriculture residues could drive into a solution high technology application. And I would like also to take this opportunity to advertise, to talk that the African Emirates that we are organizing in December 2021. I could see Prof. Paul, who is one of the panelists who already will be attending the talk. Thank you once again for your commitment and engagement for African Emirates Material Research Society. We held here in Dakar. As I will be the main organizer of this, I could be reached through this email, as you could see. So I would like also to once again to thank all my student collaborators and all people that have been involved in this exciting work that could really enhance the uses of material, especially using agricultural residues, as it is our economy is fully based on agriculture. Thank you once again.
Thank you very much, Professor Balla. I really appreciate that inside of a talk. It's always exciting to see good work happening in Africa. I have one question for you, and the question is from Jin Ho from NJU, and the question is, thanks, Balla, for your wonderful talk. Is this vanadium pentoxide-based material biodegradable?
Okay. The question is that if this material is biodegradable at the end of the device, we could say not really, as the device that is one of the aspects of the work of the development of these different devices, electronic devices and batteries, supercapacitor, and so on and so on. But what we have been mainly interested is how to develop the process that could reach into the development of this device, could be a green process. That's exactly what we have been. But after the project, after the device, we weren't really working into that aspect, and we believe that it's still not biodegradable.
Okay. Yeah. Very good.
This is where bioelectronic, as the talk of Deblina, are really one of the interesting work. And I would also take this opportunity to thank him for his wonderful talk.
Very good. So at this point, I want to invite our panelists. Professor Alice from Peking University. And then she doesn't need introduction. I think we all know her. She's a very well accomplished scientist. We have Professor Jagdish from Australian National University. And then we have Professor Paul Weiss from UCLA. And they will be discussing about this award. Welcome, panelists.
Oh, great talk. Yeah, actually I really enjoy all the talks here. So Deblina, as always, yeah, my super girl. So yeah, so glad to see you got such big contributions and move ahead. And I'm thinking maybe I first, go to Balla, ask a question for Balla. Yeah, so Balla, I saw your resume. You got 2 PhD, right? One in English, one in French. How hard it is?
Yeah.
Can you tell me how you get it?
Yeah, a very interesting question. So, here in the French system, we're having in the past we were having two PhD, what we called a doctorate of the third cycle, if I could translate word into word. So sorry. So, and after that, before reaching into a full professor, you could have what we call a doctorate data, which is a doctorate coming that being after you can be a lecturer at the university for a couple of years, but for you to become a professor, you should have that second doctorate. And when I started to have my first doctorate in Senegal, I have the opportunity also to go to South Africa, as I have been funded by the African Laser Center. And when I reached there, I said to myself, okay, even if I finish this doctorate, I will be also asked to go into the second one. Why not to take the both of them at the same time? And that's how I get into register at University of the Western Cape. And then I have been working for 4 years in the two projects. Parallelly, Prof. Maza was my supervisor from South Africa, and Professor Bay was my supervisor from here in Senegal. So I defended the first one in 2009 and the second one in 2010. So you could see that I was working into two projects completely different at the same time. Thank you very much. Yeah.
Okay, that's really surprised me.
Yeah.
So Bella, this is like the habilitation which people talk about in Europe.
Yeah, it's the same one what happened in the French system at the on the. It's just a habilitation for you to be able to conduct research or to supervise the student. And now it's cut off to 5 years ago. It's no longer being needed now. Yeah.
Okay.
Yeah.
Well, congratulations to both of you for
receiving
the iCANX awards, and then as both of you gave great talks, Deblina and then Balla. And so I think let me ask Deblina a question about this your synaptic cleft and then communication between them two, and then alignment of these protein molecules. Did you see any differences between the healthy neurons and then the people with Alzheimer's and other diseases? Have you looked at any differences, or you only looked at the healthy neurons?
Yeah, for the synaptic alignment, as per se, we have not looked into the Alzheimer's, brain yet, or neurodegenerative diseases. We have looked for the Alzheimer's. We focused on these misfolded proteins, but what we have, looked at, and along with our collaborators, also between long-term potentiation and without potentiation. So in brain, these memories are formed, and the synaptic weights basically increase, and there's this thinking that neurons that fire together, wire together, so their connections become stronger if two neurons connect to each other. And many researchers around the world are actually trying to emulate that in electronic devices called neuromorphic computing. So one interesting thing is that when there is this potentiation, that is, the synaptic connections become stronger, it seems that this alignment and this correlation between these nanoclusters actually improves. So you see more, the nanocluster size become denser, and you see that alignment improves. While on the other case, where the memory is broken, you see that these proteins become more scattered. So this is basically another mechanism of potentiation in the brain that you have found.
Yep. Okay, that's very interesting. And so we have been growing some neurons on nanowires, and then we are seeing the correlated and synchronized activity. And then maybe I'll send you some papers, and we'll talk about it later. Anyway, great work, very nice work, and then really beautiful work. I enjoyed it.
Thank you very much.
And, so in terms of your electronics one, and then have you looked at trying to really have your source also if you want to have a 2D material, so that you avoid this 3D, 2D interfaces, and then all these interface state issues and other things, so that you got a van der Waals materials on both sides of this, your MOS2, for example?
Yeah, so 2D material, the problem is that, I mean, it would be possible down the line. Right now, it's very difficult to have a very high doping, in the 2D material, because we basically want the electric field that we get in the source to channel junction should be very steep. One material should be highly doped, basically no electric field drop in that region, and you go to the channel, all the electric field drops in that atomically thin region itself. So doping the 2D materials has been a problem. What we have found from our temperature dependent studies is that, the defect states that we get in this tunneling junction was not high enough to, influence our on-state current. It might be down there, which was in the noise levels. We were not, bothered that much. But what is more important is for the electrostatic control is that the interface between the channel and the gate dielectric, that has to be, extremely pristine, because that controls, how much voltage we need to apply to increase the current. If there is in a single defect, that's going to kill it. So that interface, if we can make like 2D dielectric, that will be, very, important for the transistor design.
Yes, for example, hexagonal boron nitride, which is a large bandgap material, which could potentially act like a dielectric. Yeah, that's good. Very nice.
Exactly.
Thank you. Balla, I have a question for you. And again, so you have transitioned from one country to another and all in Senegal, and then how challenging it's been in terms of establishing facilities and be able to really carry out the beautiful research which you're carrying out in Senegal. You mentioned about challenges in Africa, about even light, because I studied in front of a kerosene lamp till I finished my year 7. So I know all the challenges, and I just want to know how challenging it's been for you to be able to establish the facilities and then be able to really carry out the beautiful work which you're doing.
Thank you very much, Prof, for the question. Yeah, the first challenge was for us as we are having a French-based system. The first challenge was the language, and it was really, honestly, very difficult when I reached in South Africa. I could not even speak a single word in English, and it was really a challenging aspect. But luckily, the supervisor that whom I was working with was speaking French, and that is how I going to be student up and down up till to that I start speaking and writing the English, and I have also the opportunity to go into a lab that there are a lot of people that are coming in and out, visitors from the US, from Japan, China, all around the world. And this open it an opportunity for us, which we really jump on it, because it was an opportunity that we said ourselves, oh, okay, this is an opportunity, we must not miss it, we must jump into it. And that is how it opened us the way to work hard and to publish in high impact factor journal. And if we see a call, we try to write for proposal, and that's how we lead into this Future Leader African Independent Research Program. And we have been funded by the Royal Society of London, and that way we get into setting up a fully operational laboratory here in Dakar, and that's how we end up by doing this work, especially here based in Africa without going anywhere. But also we managed to, with those people who have been coming in and out, where I was a postdoc as well as I was a student, I keep into collaboration with those people, which is showing another aspect of being in collaboration with scientists, best scientists in the world, also open the opportunity for you to do the work that you would like to conduct. So being in Africa or being in Europe, for me, it's not an excuse as currently, because the science doesn't have a border. Science doesn't have a border. It's just to put the energy that you need into it, and you end up by being there. Thank you. Yeah.
Very good. Good. Keep it up. Great work.
Thank you very much.
Oh, Alice,
you were muted.
Go ahead, Paul.
Yeah. Oh, okay. Very good. So maybe I'll continue along that line with Balla. So you have this advantage in Dakar that it's fairly straightforward to get in and out. And thank you for plugging the African MRS meeting that we'll have in December 2022. I wonder if you could comment on what makes a good collaboration for you. You've established all these collaborations around the world, and what is it you look for, and how do you what do you attribute to successful collaborations versus perhaps others that don't work quite as well?
Okay, so for me to have a successful collaboration is that one of the best things and that we succeed to do is that the person that I'm collaborating, I'll always have a good student that link us. And that good student is really a key parameter that helping us to really move things forward. For example, if I look at what is happening currently between me and the INRS, I'm having 3 of my students that are really registered here in Senegal that are completely working in there. And I keep on monitoring them in distance. And this is one of the key things that really put us on the spot. But it is really difficult to build it, as it's not easy to attract money, especially if you are on the African side. Writing proposal to attract money is not that easy. But if you succeed to have a good work that have been published, that you could show that I'm capable of doing this and being in Africa, so it opened the way, it opened the confidence for people to put money into you. Yeah. I think that is the key aspect.
Thanks very much. And Professor Sarkar, Deblina, I have a question for each part of your talk. So maybe let's start with the expansion microscopy. I'm wondering if when you do physically separate the material, if you think there may be artifacts in that, or if there are data that show that the expansion is smooth. We actually had Ed give a talk a few months ago, but we didn't get a chance to ask him too much. I think he was running off to another conference.
I see. Yeah. I think that's a great question. Yeah, we did actually a lot of characterization to make sure this expansion is isotropic in nature and you do not get error and artifact just from the expansion itself. So a few things that we did was, first of all, we used stereotypical structures, for example, microtubules and other structures for which the nano-organization is known from other studies like EM. And from doing these structures and performing expansion on that, and you see that the orientation and everything is what we should expect from previous literature. We are also trying out different kinds of man-made synthetic structures in which we can design, say, protein origami as well as DNA origami. So the exact location between proteins and biomolecules, and so we can know that if it was a triangle before and it doesn't become into a circle, and those kind of the relative distances are maintained because we design it ourselves. So that's another thing. And another thing we are doing is we basically take a super resolution image of a structure before expansion. And of course, those super resolution imaging technologies have the other challenges, such as you cannot scale to the whole brain. They only work with multiplexing is difficult, but for our validation purpose, we can we took, like, there is a DNA paint imaging. Also have done storm imaging, which also gives like 20-30 nanometer resolution. So we take an image of the tissue before expansion using those super resolution technologies, and then we do our expansion procedure and take image of the same region after expansion, and then we can actually do processing to shrink down the imaging and compare and register. And you have found that the errors are actually less than 2 to 5% in nature, up to like 20-fold expansion factor. So yeah, we have done a lot of characterization to make sure we're not introducing errors due to expansion.
Very good. And in the device part, in your the early part of your talk, to what extent do you consider the ability to fabricate and ultimately to manufacture when you're developing a new potential device to lower power or shrink the scale of the devices and all the other goals that you had for those new structures.
You mean large scale manufacturing?
Yeah. Like, would these materials be compatible with a fab, or would there be a way to put them together? In reality, when you put billions on a chip that you put in your computer.
Yeah, actually, a lot of work needs to be done till we go to that stage. So first of all, this 2D materials, just the growth itself, we can grow. For example, we have like right now we can grow wafer scale graphene and industrial scale, do roll-to-roll transfer. For other 2D semiconductors like molybdenum, one that I used was still like we could grow wafer scale. Actually collaborated with a group because I'm not a material scientist, but I think we have come a long way right now with 2D materials. It should be possible to do wafer scale, and the transfer can also be industrial scale in nature. Do we need further improvement? But the vision is really that you should be able to, like in academic labs, we can build, with our limitation, builds only few devices. I mean, not few, still hundreds of devices, but not say billions. And show that technology is promising, and then that it up. And with the 2D materials, the technology as it is advancing, so it should be possible to do industrial scale billions of devices and have them in our computer, one day.
Very good, thank you. Maybe I'll turn it back over to Martin.
All right. A very quick question for you, Balla. I think the infrastructure in Africa has significantly changed just a few years of being away. How do you see the landscape in terms of the traditional wet chemistry that dominated research in the 80s and the opening up from materials research in Africa? How do you see that balance playing out in the continent?
To honestly speaking, people are still doing this traditional chemistry synthesis or preparation of material in general. But if you look at currently what is happening in the continent in terms of synthesis of nanomaterial, we could see that besides South Africa, where we have a very active group that are using biosynthesis method, I could see that it's people still working on traditional aspect of synthesis of material. Most of them. I know only two groups in South Africa which are really using this green synthesis method. Otherwise, the rest they are still working on traditional chemistry, wet chemistry synthesis. Yeah.
Well, then it calls for more partnership and opening up the talent in that continent, because it's very amazing to see that you can do very quality SEM there. I didn't have one in my university when I moved there.
Yes, you could see that most of research also is still concentrated in the southern region of Africa. West Africa, not really. Beside Nigeria and Ghana, you could not see some instrument on that level. Now, most of us, where you need to access some specific instrument, you go to South Africa. That's why most of us are making collaboration with South African scientists. And we have also North Africa, in the Maghreb region, where Morocco, Algeria, they are doing the best. Even what this country, Tunisia also, they are doing the best. But the most science is still concentrated in the southern region, especially South Africa. Yeah.
Okay. Very good.
If you take, for example, West Africa, beside Senegal, Nigeria, and as well as Ghana, I could not say any country that are really involved into research. And if you take Senegal, only Senegal, I could say there is none also beside what we are trying to do ourselves. Yeah.
Very good. Thank you very much. Congratulations to both of you for being the inaugural Young Scientist Award. I hand over to Aris.
Okay, yes, that's great. Actually, I want to give the last chance to congratulations to Paul and Jagdish. As in iCANX stage, long time we really put a lot of efforts to pop up for the young generation. I think last year, Paul, you rising for the, ACS Nano, the Rising Stars. We have Martin on the stage at that time. Now Martin is already with us. So Paul, did you have any words to, say to Martin?
Well, Martin, we're delighted to have you with us. We appreciate your wisdom at the journal and all your support. And now, we look forward to counting on you here and to working even more closely with you.
My pleasure.
Okay, Professor Jagdish. Yeah, I think you really done a lot to, to pop up all this young generation. Did you have any words for three of them?
Oh, well, I say that, you are all absolutely rising stars. You all three of you have got a great future ahead of you. And then, and I think Balla has pointed out the collaborations and how important the collaborations are, because we cannot be experts in everything. By collaborating, we can really be able to really do much exciting science and also much more comprehensive science. And I do hope that you'll make use of those collaborations and also try to reach out to the people, those who do not have the facilities. For example, Deblina being at MIT, and then having also started her life in India. So then there are many opportunities to collaborate and enable the careers of other young scientists in other parts of the world. So really, for me, the most satisfying thing is to see the young people being successful. And in fact, my wife and I have started an endowment at the ANU to be able to attract the young scientists and students from the developing countries to come and spend some time at the ANU, because of the fact that the opening doors for the young scientists from the developing countries is absolutely critical, because there's so much of talent there, but they don't get many opportunities, and it's important for us to open the doors for them so that we can make use of that talent for the good of the world, good of the countries which they come from as well. So congratulations again. And we hope that you'll get involved in the future also in the iCANX talk series. And then who knows, one of these days you may become an organizer as well. We look forward to your contribution to that as well. And good luck to both of you in your careers, three of you in your careers as well.
Okay, great. Thank you. Yeah, Professor Jagdish and Paul. Yeah, actually, I'm so proud of you to work with all of you. This was a wonderful platform, and I think this was wonderful. This time we have a brand new blood here. Yeah, so we have Martin. Yeah, as I said that last year we met Martin on this stage, as today we met Deblina and Balla. Exactly the same. We want to see the new face, to share the experience and everything. Now we already, have Martin as our partners. And later we will see you almost every month once at least. Yeah, you also can, contribute a lot to invite the young scientists. And Professor Deblina and Balla. Yeah, so as the first, two young scientists to win this award. I say that, this was a message I want to deliver to the whole world. As no matter where you are, no matter what are you doing, if you're doing good science, if you are, want to connect with the world and universe, iCANX is your stage. Yeah, you are welcome to join iCANX. I also want to thank to all these people here and all the people who was behind the screen. We have support from Applied Physics Review. That's Professor Jagdish was the editor, in chief, and sponsor this. And ACS Nano, Paul was the founder of this journal. I support this as always. Light, Science and Applications, they are super journal, and they support all this young science, award too. And OAE is a publication group. They have several journals like soft science. I was on the edit board, so yeah, I was trying to help all these young scientists award to happen, and I want to invite more young scientists to join us. Now, Deblina, Sarkar, you have this beautiful certification. Yeah, proud of you.
Oh, great. Thanks so much,
Yeah.
Alice.
It
Thanks to
is.
iCANX.
Balla, you have this certification too? Yeah, if we met on site, I will deliver hard copy to you. Now I deliver the e-version.
Thank you very much.
You're welcome. Also, this was the first time I said, now I send out the call. Yeah, we call for the iCANX Young Scientist candidates. If you are qualified as this, you know, if you are get your PhD degree within 10 years, now you're okay. Yeah, already qualified. Please provide 5 materials. Yeah, to us. Yes, is your full CV and the science contributions and a brief research plan and maximum 5 respected publication or patents and 2 recommendation letters from the senior scientist. So you send to us to the email with the info at icanx.com. The deadline for next, Young Scientist Award is for October 10th. So please do submit your material to us. We are welcome all of you to join us. Okay, yeah, that's today, that's tomorrow. Next week, we will have Professor Liu Jing to talk about the liquid metal science and technology from fundamentals to industry practice. So we're looking forward to meet you every Friday on iCANX Talks. Thank you very much. Thank you for staying with us. That's all for today and see you next week. Bye.
Okay.
Yeah.
Thank you, everyone.
Thank you.
Bye-bye. Thank you.
Bye-bye. Very great.
Bye.
Congratulations.
Thank you.
Congratulations
Paul,
again.
you can see.